Thank you very much for the introductions, and thank you, ladies and gentlemen, for allowing me to speak on behalf of the company and present Exothera and Exoready to you. So how does it work? Voila. In a nutshell, Exothera is a virus-focused CDMO located in Belgium that is focusing on providing our clients a one-stop shop experience, and you can see that we are covering quite a significant range of all the activities that our clients might need throughout the cycle of their process and product development. And we are operating 2 state-of-the-art facilities located roughly 45 minutes away from Brussels. In a nutshell, speaking about our track record, so a significant part of our pipeline focuses on 2 modalities, on adeno-associated virus and on the adenovirus. We do have quite a significant track record with a few key cell lines like HEK, A549, Vero, and others. We are very proud to say that we have open collaborations in AAV domain with companies like Remedium and Logic Bio, and we have collaborations in the adenoviral domain with companies like Rocote and Reitera and others. And this all comes together with the consistently high client satisfaction rate, which we are quite proud of as a CDMO. So today I would like to really focus on a bit technical side of our offer and walk you through the key services that we offer for upstream, downstream, and analytics, speaking of helping our clients to fast-track their adeno-associated virus to clinical implications and beyond. Speaking about the track record with AAVs and such, it's very important to highlight that we have clients that are coming to us with products that are historically developed in 2 different modalities of the cell lines, adherence and suspension. And we do provide a technology solution, a platformized technology solution that could be very well fit to both. We've been recently publicly disclosing the results of scaling up our runs to 2,000-liter vessels with 950 working volume with the consistency of titers across the scales, and this would be also something that we would cover in the upcoming next slides. And you can also see that till date our major experience resides with 4 different serotypes of adeno-associated virus. So from now on, we'll speak a little bit more about the technicalities of the platform that we offer to our clients. And the platform as such is called Exoready. Apologies for the intensity of the slide. Please don't try to read it. It made blank intentionally. But if you do have any questions, please talk to us later. So Exoready essentially consists of 9 unit operations throughout upstream, midstream, and downstream sections of the process, allowing us to really fast-track the experience of our clients on the very solid CMC foundation that includes quality by design, criticality assessment, and design of experiments implication. And in this talk, we'll cover a few case studies with the key unit operations like bioreactor capture and polishing. And you will see that for each unit operation, we offer a set of pre-fitted parameters that we are optimizing for our clients, as well as the set of appropriate analytical controls. The 2 cases in upstream that I would like us to briefly go through include the case A, which is entirely focused on Exoready, and Exoready again is the platform that we are offering, and the case B that discloses the public collaboration that has been announced in 2022, and that includes works that we've performed with Logic Bio and Polyplus in terms of scaling up the transient transfection process using the HEK cell line for production of AAV serotype 8. We'll focus a little bit more on the application of design of experiment at Exothera and the benefits of this statistical approach in development. Excuse me. So in case A, we pursued the task of tightest optimization using a small-scale model, and we've been using a high-throughput small-scale system in upstream to identify the parameters responses relationship, and we really used a multi-block design of experiment to make it happen, ultimately identifying four statistically significant relationships and preserving them as key parameters to maintain throughout further process optimization. And we would see the impact of these on titer in the next slides. In case B, which is a logic bi and polyplus case, we've been pursuing some kind of a different purpose. We had a process that has been originally pre-developed by the time we had our hands on it. And we were really investigating the key parameters robustness when scaling this process up to 2,000-liter bioreactors. Again, identifying the key relationship between titer response when it comes to varying the parameters like power to volume, transfection volumes, and other inputs. And so here you can see a nice design of experiment layouts, but we will see their translation into the results on the next slides. So this kind of systematic statistical approach allows really to gain a very significant and very detailed process understanding that we are offering to our clients in terms of the pre-fitted responses. And when looking into the results of this and their translation into larger scales, one can notice that actually the performance across the scales appears to be quite homogeneous in terms of the key, let's say, parameters. And here we are looking into the four different charts across the board of 6 parameters, and essentially we are looking at the scales of 1 liter, that is our high throughput DOE optimization scale, 40-liter bioreactor, that is our largest non-GMP scale, and then 200-liter and 2,000-liter vessels, which you can see perform quite consistently, at least in terms of the metabolite profiles that we illustrate here. However, more important than this is probably this slide, and here we see two different visualizations for 2 different types of processes that we are currently having in-house, and it speaks quite well about the approach we use to tackle the challenge of scalability. As you can see here, with serotype 8, using this statistical approach, we were able to secure the parameters that allow us to have our titers in the range between 6 and 8, 10 to 11th viral genome copies per ml, which is quite a high-end result for the HEK transient transfection process that we've been running, which is, of course, something that we are very proud of. And to speak about what happens further, I'd like to develop a few key messages about experience with downstream and analytics, as it is also very key, and I'd like to speak about the way we approach capture, polishing, and some analyticals work around this. So the capture solution at Exothera is really based on affinity chromatography, although we do have ways for clients that have a bit unique serotypes. And we largely work with 2 types of resins, which you can see on the first graph have very different properties in terms of binding and elution profiles. We work a lot with elution optimization and adding elution modifiers, as you can see on the image 2 and 3 to the right, which allows us to have a kind of a standard toolbox and really approach with versatility the questions of elution completeness and stability, and also the washes for this step. Furthermore, this platform approach includes the cycling studies that we do for the resins of choice, and the first graph illustrates the consistency of the recoveries and consistency of host cell impurities clearance, which is quite important for us across the processing, as well as consistency of the process chromatograms. And the last graph is really speaking on the recoveries for a given serotype, and it's fair to say that typically starting with non-optimized recoveries around 50–60%, we end up with recoveries around 80 and more, but of course this is largely serotype dependent. And last but not least, maybe the most interesting part, so this is approach to polishing, and we offer a convective media-based standardized polishing approach. And you can see here in the column format the experience with two different serotypes. And we think it's quite remarkable because really on the left you can see that standard approach really works quite well for the chimeric serotype, and addition of a simple elution modifier on the left allows to further shorten the gradient and increase the resolution at the same time. Whereas on the right, we've been working with the serotype 2, which is generally a bit more challenging. And you can see that the standard solution does not really result in much benefits. The purification is literally not there. However, it's not really this unit operation optimization that allows to achieve more, but the changes that we apply further upstream of this step, in this particular case, changes to bioreactor parameters and changing to affinity chromatography step that allows to simplify the experience with polishing chromatography, which I think is a good message, as very often the unit's operations are interdependent. And indeed, it's key that chromatography development for viruses and viral vectors is coupled with appropriate analytical techniques. And in our case, we rely a lot on the use of HPLC chromatography, anion exchange HPLC actually with fluorescence detection. And here you can see that this is a rapid and cost-affordable way to really screen in a high throughput setting across the fractions that we generate. And on the right side of the slide, you can see that the method development and method optimization is also based on the DOE and specific gradient optimization, which allows us to, in the end, have a very significant resolution between the peaks. I think it's also very important and fair to say that, of course, the analytical approach at Exothera will be orthogonal and proportional to the need, and also this method is quite easy to accommodate across the different serotypes as needed. And so in the summary, the takeaway messages for me are really the following. We are very happy to offer our services and secure the high-performance upstream process with consistent titers across different scale, and couple it with the very significant enrichment, scalable purification process, and to ultimately envelope all of it into the process and CMC package that allows to fast-track the product development from 9 to 12 months roughly to reach the clinical milestones and beyond. Thank you very much for your attention, and if you have any questions, please let me know, and please stop by our booth, which is just right there if you would like to reflect on this and then ask your question. Thank you.